Hello students, today we will be starting our first lesson in the cell structure and function, and this has various subsections like the cell and the unit of life, where it is subdivided to cell envelope, endomembrane system 1 and 2, biomolecules where it is included carbohydrates, lipids, proteins, nucleic acids and enzymes, and the cell cycle and cell division as a third subsection. So today's objective of the class is we'll learn more about the cell envelope and the endomembrane system. This is a short table of content that we intend to cover in today's lesson, and you can have a look, but we will cover these things in today's class. So coming to what is cell biology, okay? So first of all, you close your eyes and picture a brick wall. What is a basic building block of that wall that you see? A single brick, right? And like a brick wall, your body is also composed of basic building blocks, and the building blocks of your body is cells. So they are living things. So all the organisms, living organisms, are made up of cells. In simple words, the cell is the simplest entity of matter that can be considered as a living entity. Even when these cells are arranged into higher levels of organization, because there is not a single cell that we can see in daily life moving here and there, right? Though in bacteria they do, but we cannot see them through our naked eyes. So they are actually higher organisms, and they have a higher level of organization through tissues and organs and organ systems, and cells remain the organism's basic unit of structure and function. Okay, that's very important to remember. Now if you see the unicellular versus multicellular organisms, like unicellular means a single cell, right, which can live on its own, just like this is the example of paramecium. The size of the paramecium is around 40 micrometer. We cannot see even through a light microscope. We can observe only through the electron microscope. And this is an example of multicellular organism systems, and where they are the basic building blocks cells, of course, but they have higher organization systems like tissues, organs, and organ systems. So this is an example of the size range of various cells, which will give you an idea what is and how they look like. For example, atoms, which we have learned in chemistry, they range from 0.1 nanometer. 1 nanometer is 10 to the power minus 9 meter. That means it is less than 10 to the power minus 9 meter. And so no chance of looking them through even an electron microscope, which is a very powerful microscope. Powerful microscopes, we can see the proteins and lipids, and viruses, or the smallest bacteria or smallest cell, it is 100 nanometer. So even here we cannot see through light microscope, the microscopes which are present in the laboratory. So we cannot see the bacteria through with the help of them. We can observe only the bacteria through the electron microscope or higher resolution microscope. Okay, so the bacteria, whatever we carry, for example, when our hands are dirty, so the bacteria or the viruses, we cannot see them through the light microscopes, and no chance with the naked eyes. Here there are examples of plant cells and animal cells which we can observe through light microscopes, and the microscopes which are present in the laboratory. This is the human egg, which is 100 micrometer in size, which we cannot see even through naked microscopes, but we can see through the light microscopes. And here is an example of chicken egg, which are much bigger in size. We may eat it, right? But here it is only made up of one cell. Length of some nerve cells and muscle cells vary from this range. So we can see through the naked eyes without the help of microscope. So this is just a brief idea how the cell looks like, so that you can understand. Now the discovery of cell. So Robert Hooke, you all know that he coined the term cell, right? and but he observed the dead cork cells. So he coined the term cells from the word jail, which are known as cells, and he used 30 times magnification microscope. So he is Robert Hooke. Now Leeuwenhoek, he saw the first living cells in the year 1665. Okay, so but living cells, so it's important to know, and he observed the cell Spirogyra. It's an organism. And Robert Brown coined the term nucleus. He observed through the microscope, but he did not know the function of the nucleus. Chilton and Schwann, they are also famous scientists who proposed that plants and animals are made up of cells. So everything, whatever we see around, moving here and there, they are made up of cells. Virchow, he was a physician, and he proposed that healing wounds in humans, the cells are coming from cells. Okay, so these are some important discoveries in the path of the knowledge that we have gathered about the cell. Now, these are the important years that may be helpful for you to know. So it's good that if you remember them. And here there's an extra thing. Louis Pasteur, who disproved the spontaneous generation in the year 1859. He proposed that no living organisms can be generated spontaneously or by its own. They can only generate from another living organisms, for example, a cell. So he disproved the spontaneous generation. So Anton Leeuwenhoek, just now we mentioned about him that he observed Spirogyra, an organism, from pond water, and he was first to observe living cells. So it's important to understand the question. For example, the question can be, if it's living cell, who discovered living cell, then the name should come about Leeuwenhoek and not Robert Hooke. Okay, because Robert Hooke observed the dead cell from a bark of the tree, and Leeuwenhoek used 300 times magnification microscope. Okay, now do you have these recipes at home? Just joking. So people initially thought that the organisms grew from non-living materials, and this is a recipe of a mice. Like if you take dirty shirts and feet along with it and keep them for 21 days, the mice can be generated. But now we all know that it is not true, right? Mice can generate only from a baby mice, and which can be generated from an egg of a mice. So this was the thing that people initially thought, and hence at that time Francisco Redi experiment, he was the first person who proposed that living things cannot be generated from a non-living matter. Okay, so what it did, he took the non-living matter, that is a piece of meat, and he took it, two pieces of meat. One he put it in as a control group and one as an experimental group, and one of them was kept as an open and in another second experimental group, and he tightly corked the container. Okay, so his observation was that, as expected, was that for the first set of control group experiment, where there was no cork attached, it was free, the air can come in and go. Here he observed that small worm-like structure have generated on the mice with the time, on the meat with the time. Okay, so but this was not the case with the experimental group. So his observations was that the maggots, we call them maggots, come from flies, not the meat. So he concluded that it is not possible, because the maggots didn't appear in the experimental group, where the mice entry was restricted. So he concluded that life comes from life, and he proved that the spontaneous generation is not true. Okay, people didn't believe him, but he was right at that time. Okay, now this is a simple diagram of microscope you may be using in your laboratory, school laboratory. and it's important to know some of the parts of microscope, and for that reason I have shared this picture with you. So for example, this is the eyepiece. So eyepiece is where we keep our eye, and here there are different lenses, and it magnifies the image and we keep the object at this place. Okay, now coming to different types of cells. Your body has many kinds of cells, each specialized for a specific purpose, right? Just as the home is made up of a variety of building materials, right, like bricks, cement, etc., your body is constructed from many cell types. For example, the epithelial cells, or the skin cells, protect our surface body, and they cover the organs and cavities within them. Bone cells help to support and protect the body. Cells of the immune system fight against the invading bacteria and viruses. The blood cells helps in the transport of the oxygen and nutrients to various parts of the body, right? And removes the wastes like carbon dioxide. Each of these cell types plays a vital role during the growth and development of an organism, for multicellular organisms, right? And but what is common? What is the common between all the types of cells is that they share some certain fundamental characteristics. Whether you take a bacteria, or you take an onion cell, or you take a human cell, they share certain fundamental characteristics. So this is just an example. Now, like the skin cells and bone marrow cells, they divide very rapidly. On the other hand, the muscle and the nerve cells do not divide once they have developed. So they are different in functions to some aspect, but they have the basic structure and functional of the unit are the same. Okay. Now there are four main types of cells. So even though there are several hundreds cell types in the body, all of them can be grouped into just four main categories or tissues. This makes them easier to understand. Okay, so just like epithelial cells or the skin cells, nerve cells. Secondly is nerve cells, and third is muscle cells, and the fourth one is the connective tissue cells. Okay. Now, this is the section which is observed under the microscope, where actually all the four types of cells are present, if you see. So here, if it is the capillary and the vein cells, and they are the epithelial cells. And here, so the stained one, what is the purple one and purplish blue ones? They are the stains that we have seen, and we have put purposefully so that we can observe them in the microscope. Okay, so this is the epithelial cells due to the characteristic. We'll see in due course of time. This is the nerve cell, which is the largest in size. We can observe in the through the naked eyes even. This is the nerve cells, and this is the nucleus of the nerve cells. This is the connective tissue cells, which helps in the transport of various nutrients and wastes from one place to another. This is also connective tissue cells. So these are isolated. And this is the muscle cell, if you see the elongated nucleus. Okay, so if we have some idea about the categories and the functions of the different types of cells, we can identify which cell is it through the microscope. Now, important thing to know is that the largest cell in the human body is the ovum in females, and the smallest cell in the human body is the sperm. Okay, these are the gamete cells. This is we know already. And brain cell, we name it as neuron or nerve cells. Liver cells, we name it as hepatocytes, and kidney cells, we name it as nephron. Okay. Now, close your eyes and picture a brick wall. What is the basic unit of that wall that you see? We have just now exercised this, right? That a single brick we see, and this brick is just like this brick is the building block of that wall. Our cell is the building block of the whole body. So cells are the basic units of life, capable of doing all the required biochemical processes that a normal cell has to do in order to live, right? And what are the biochemical process functions? They functions like its respiration, digesting the food, getting rid of the metabolic wastes, right? And obtaining energy, of course. And cells are capable of performing all the metabolic functions of the body. Hence, the cells are called the functional units of life. So the questions can be asked why the cell is the basic unit of life. Here lies the answer. Now, functions of the cell. So the cells perform different functions as we have discussed just now, and examples of cell names are very important to understand the questions. For example, here the neuron transmits information from brain to the different parts of the body. For example, there is a hot plate and by accidentally your hand has touched there. So what the brain says that you need to remove that, right? Otherwise you may have a burn. It is also called the reflex action, but yes, some actions are directly the transmission of the information from brain to different parts of the body. And the brain cells are called the neuron cells. Nephron, on the other hand, is the cell name of a kidney, whose structural and functional unit of the kidney, which helps in the removal of nitrogenous waste materials from the body. Okay, and these cells perform variety of metabolic reactions such as the breakdown of complex molecules to release energy. So of course the releasing energy is same for both the cells, neuron as well as nephron, because it's a fundamental need in order the cell to live, right? And cell also synthesizes new molecules required by the body. So this is just a small note that may help you to understand the different organelles within the cell within a human cell especially, not a prokaryotic cell or a plant cell. So here you can understand the small functions. This is just a note like of thing that you can note it down. Now, functions of the human cell. So, if you see, the human cells are multitasking, right, when it comes to functions. So, though there are infinite functions, they perform six of them are very, very important. And the first of them is the provide structure and support. So, colon-chymatic and sclerenchymatic cells contribute to the structure in human. The cell name is the colon-chymatic and sclerenchymatic. You have learned in the chapter of tissues. And for vascular plants, the xylem and the phloem provides the structure. So these are the names of some cells. Cells also facilitate the growth during the process of mitosis, right? Mitosis is nothing but the formation of two daughter cells from one parent cells. The third function is the passive and active transport, that the cells import nutrients and export the waste materials through two types of transport. Okay, we will learn in due course of time in detail about them. The passive transport for the smaller molecules, which includes diffusion, okay, and larger molecules, proteins, through active transport. They cannot be spontaneous. They require some energy. And the fourth one is the production of energy through photosynthesis in plants and respiration in animals. Various metabolic reactions like the glycolysis, Krebs cycle, they are the different chemical processes that happens in the cytoplasm as well as some in mitochondria, which is the powerhouse of the cell, you know. And finally, the reproduction is the sixth function, which also occur in two ways, that is asexual reproduction and the sexual reproduction. And the two divisions that are important here to know is the mitosis and meiosis, which we will learn in the due course of time in detail. Now, this is an experiment of Louis Pasteur experiment, who showed that bacteria are present in the air and they do not appear spontaneously. So what he did, he prepared a broth or a liquid and he heated it for a specific very high temperature, and he left that one to be cooled, reducing the temperature, and he kept the flask open, and he waited for some time, and he found some growth, and he observed it through the microscope. So at that time microscopes were already discovered. And for the second experimental group, so this is the control group and this is the experimental group, he sealed the flask. So everything is the same, the quantity of broth is the same, the type of broth is the same, but what he did, he sealed the flask and he waited for the same time, and he found that there is no growth of bacteria, right? So what is his conclusion? He supported Reddy's research, which Reddy did it with the help of meat. And he discovered that cells must come from other cells, right? And he disproved the spontaneous generation and said that life cannot just appear of nowhere, right? So it is proved. And Pasteur came up with the idea of pasteurization. You may have heard this term. This is very, very important term, pasteurization, after discovering that bacteria could contaminate the milk from the air. So we need to heat or remove the bacteria from the milk before consuming it. And this process kills the bacteria so that it does not harm us. So pasteurization is a process of killing the bacteria within the milk at very high temperature and then cooling the milk so that it is bacteria free. This process is also used in making the cheese, yogurt, etc. in large scale, in industrial scale. Now coming to the cell theory. The theory states that all living organisms are made up of cells, which are the basic unit of life. Okay, so in fact the actions of organisms are all based on functioning of cells. For instance, the movement of your eyes as you read the sentence results from the activities of the muscles and nerves, right? And even a process that occurs on a global scale, like the recycling of the carbon atoms, is the product of the cellular functions, including the photosynthetic activity of chloroplasts in the leaf cells of the plants. So who discovered the cell theory is very, very important. It was discovered by Schleiden and Schwann. So Schleiden was a German botanist, and he, along with Schwann, they proposed, who was a zoologist. Zoologist means dealing with animals, and botanist means dealing with plants. So they came up with a theory that all the plants and animals are made up of cells. So the three most important points that we need to remember in the cell theory is that every living thing is made up of one or more cells. Cells carry out the functions needed to support life, to make it a living entity, right? And cells come only from other living cells. It cannot be spontaneously generated. So this is an example of worksheet that I will advise you to note it down in your notebook and complete it based on your knowledge till now. Okay. Now, to take a moment to look at yourself. Okay, stop for a while. How many organisms do you see outside your window? Your first thought might be that there is just one, yourself, especially during this situation. However, if you look closer at the surface of your skin, or even inside or think of inside your digestive tract within your mouth, you will see that there are actually many organisms living. Okay, and that's right that you are at home, but you are around a surrounding with trillions of bacterial cells. So they are living entities. And this means that your body is actually an ecosystem, just like an ecosystem. You need not go somewhere to find an ecosystem. Your body is the biggest example of an ecosystem. Okay? All cells fall into one of these two broad categories, the prokaryotes as well as eukaryotes. Only the single-celled organisms of domains like bacteria and archaebacteria are classified as prokaryotes. Pro means before and kary means nucleus. Okay, so before nucleus, so even nucleus was not discovered during that time. Animals, plants, fungi, protists are all the eukaryotes. Eu means true. What is the meaning of eu? It is true and are made up of eukaryotic cells. So these are true cells. Often though, as in case of we humans, there are some prokaryotic friends hanging around. That is, we as a human body have a small ecosystem, right? So, question to think is that, or are all the prokaryotes single celled? So think about that. Prokaryotes means bacteria or archaebacteria. Are they single celled or not? So actually, in general, prokaryotes are single-celled organisms, right? Just like bacteria. However, there is an increasing evidence that some groups of prokaryotic organisms or cells can organize to form some structures like this that resemble the multicellular organisms. Whether this counts as real multicellularity or is this a question, of course. Okay. For example, this is a cyanobacteria picture. They form the long filament-like chains, and in these chains, the cells remain connected to each other after the division and acquire unique cellular identities and function. They just behave like a multicellular ones. Some cells in this chain are specialized to carry out photosynthesis, the production of sugars using energy from the sun. Okay, and these are the smaller, darker cells. These darker cells, whatever you can see, that make up the most of the chain, and others are specialized to fix nitrogen, converting atmospheric into the biological useful forms. So in this case, is an example where the prokaryotes also behave like the multicellular ones. So this is just an example. So this is a question to think. Okay. Now, the basic components of prokaryotic cells are plasma membrane, right? Cytoplasm, DNA or RNA, mostly RNA, and ribosomes. Okay, so they are some components of prokaryotic cells. So the plasma membrane is an outer covering that separates a cell's interior from its surroundings, right? And cytoplasm, which consists of a jelly-like substance like the cytosol inside the cell, plus the cellular structure suspended. And in eukaryotes, cytoplasm is specifically means a region outside the nucleus. So the definition of cytoplasm is slightly different for eukaryotes and prokaryotes. DNA or RNA is the genetic material which carries out the information of the cell, and ribosomes, which are the molecular machines to synthesize proteins. So these are the 4 basic components of the prokaryotic cells. Okay. So despite these similarities between prokaryotes and eukaryotes, but actually they differ in a number of important ways. So this is an example of prokaryotic cell, which is very simple. It is a single-celled, mostly organism that lacks a proper nucleus. Okay, and they also lack the membrane-bound organelles. The prokaryotic cells are not divided up on the inside by the membrane walls. Like, for example, they have ribosomes, which are not membranes, and other organisms, other parts organelles also, they don't have a membrane-bound structure. So the majority is found in a central region of the cell. We call it as nucleoid. So this region we call it as nucleoid. They don't have a proper nucleus as well, and it typically consists of a single large loop. We call it as circular chromosomes. Okay. So in normal eukaryotes, they have a double helix DNA or a structure, but for bacterial or prokaryotes, they have the circular chromosomes, the nucleoid, and some other frequently seen structures of features of the prokaryotic cells are in this figure, which is actually an example of the actually rod rod-shaped bacterium. So even bacteria are of various shapes. This is an example of rod-shaped bacteria. Okay. Now, most bacteria, however, surrounded by a rigid cell wall. We call it as which is outside the membrane. We call it as peptidoglycan. It is made up of peptidoglycan, and the peptidoglycan is a polymer composed of the linked carbohydrates and small proteins. So this is the peptidoglycan layer, and peptidoglycan is a polymer which is contains sugars as well as amino acids, and they form a mesh-like structure. So this is a meshy-like structure. If you see here, and the plasma they are present outside the plasma membrane. Okay, and the cell wall provides an extra layer of protection which helps in the cell maintaining a shape, its proper shape, and also it prevents the dehydration. So the cell wall's function is here. And many bacteria also have the outermost layer of carbohydrates. They are called capsule. So outside the cell wall they have a capsule, and which is very sticky in nature and helps the cell to attach, okay, to some environment or where it can grow or reproduce. This is just an example of different types of two bacteria which just may be useful for knowledge. Like this, you can see that peptidoglycan, one, it has a thick peptidoglycan layer and another has a thin peptidoglycan layer. One is called gram positive and one is called gram negative, and they behave differently. We will learn about in due course of time. Okay. Now, some bacteria have a very specialized structures around found in the cell surface. So this is the cell surface, which may help them to move or stick to the surfaces or even exchange the genetic material with other bacteria. For example, this flagella are a whip-like structure and that act as a rotary motors that to help the bacteria to move. Okay. And the fimbriae, so this is the example, and it is the proteins made up of flagellin, and they are present in almost all bacteria. Okay, and the fimbriae, which is a numerous hair-like structures that are used for attachment to the host cells and other surfaces. So if you can see, they are shorter than the flagella, right, in length. And another type of structure, we call it as pili, which are longer, but they actually they come in different varieties. For example, some types of pili allows the bacteria to transfer the genetic material. So this pili helps in transferring the genetic material. So you can see the difference in pili and fimbriae is that the fimbriae is function is mainly to attach to a surface. Main function is the attachment, but here the pili are used for attachment as well as the genetic transfer. Also, the chemical composition are different. This is pilin and this is fimbrilin. So the chemical composition is also different. Now here archaebacteria also may have most of these cell surface features, but their versions of a particular feature are typically different from those of the bacteria. So they are both prokaryotes, but they have some differences. The cell walls of this archaebacteria are actually distinctive from those of eubacteria, which are a true bacteria. And archaebacterial cell walls are composed of different polysaccharides and proteins with no peptidoglycan. So they have peptidoglycan and they have no peptidoglycan. And many archaebacteria have cell walls made up of polysaccharides. So actually archaebacteria is called the linkage between the prokaryotes and the eukaryotes. So it's a very important part. They have some features common to bacteria and some very little to eukaryotes, but they are categorized under the prokaryotes. Okay. Typically, the cell size of the prokaryotes are far less than that of the eukaryotes. So the cell size, if you see here, The prokaryotic have 0.1 to 5 micrometers, which is significantly smaller as compared to the eukaryotic cell. Okay, so what is a eukaryotic cell? So first of all, what would be like if you live in one room cabin? Well, things would probably be pretty simple, right? And you would eat, sleep, work, relax in a single room, which may be creepy. But prokaryotes are very simple cells of organisms, just like bacteria, but are sometimes compared to one-room cabins where they perform everything within one specific membrane, that is the plasma membrane outside, and within the cytoplasm there is no membrane at all. They don't have an internal membranes, so they are just like the single room with no walls to guard it up, right? But eukaryotic cells are more complex than the prokaryotes. So just as the large family home split into many rooms with different purposes like bedrooms, bathrooms, kitchens, etc. And so eukaryotic cell also, they have different functions. They have different specialization functions of different organelles, and they are neatly separated by another layer, by layers of membrane. So this organization lets each compartment to maintain its own conditions. For example, the lysosomes. Lysosome is an organelle in eukaryotic cell, but not in prokaryotic cell. They act as recycling centers, okay? And they must maintain an acidic pH, that is acidic environment, in order to dispose the cellular waste. Similarly, the peroxisomes is also another organelle in eukaryotic cells, which carry out the chemical organized reactions like oxidation reactions and produce the hydrogen peroxide, okay, and both of which could damage the cell if they weren't safely stored away in their own room. So they should be there in the specific room because their function is very discrete. Okay. Now this is just an example that of eukaryotic cells and with proper labeling, and here we can see that the compartments are lysosomes are there, Golgi bodies, ribosomes, nucleus, nucleolus, and each have a specific membrane. This is endoplasmic reticulum, smooth and rough, and so it's a big part of the reason why eukaryotic cells can grow to be many times larger than the prokaryotic ones. So they are larger in size than the prokaryotic ones because each one has a different function and they have a different rooms with separated with the membranes. Okay. Now this is just a Venn diagram which shows you some similarities and some dissimilarities between eukaryotes and prokaryotes. Like they have similarities, like both have flagella sometimes, plasma membrane, cell division occurs in both the cases. They have both cytoplasm, ribosomes and chromosomes, right? But what's the difference? That eukaryotes are far bigger and prokaryotes are smaller. Membrane bound nucleus and nucleolus is there. Here we call it as just circular chromosomes. We call it as nucleoid for prokaryotic cells. Complex appendages. Appendages means the process of sticking to a surface. It is very complex for eukaryotic cell, but it is very simple with the help of pili in prokaryotic cell. The chromosomes are also different. Like here it is DNA, which is linear along with proteins. We call it as histones. And here for prokaryotic it is circular in shape. Okay, membrane bound is the major difference. Also the process of cell division, that is mitosis as well as meiosis for gametic cells in eukaryotes it happens, but for prokaryotes it happens with the help of very simple binary fission. Okay, so the process of cell division is also simple in prokaryotes but complex in eukaryotes. Cell wall is present in plant cells, whereas absent in animal cells. So it is a complex. So this one is complex peptidoglycan. We have learned that it's a complex cell wall for prokaryotes. Cytoskeleton and big ribosomes are there for eukaryotes, but the ribosomes shapes are very small for prokaryotic cell. Now coming to the parts of the cell. This is the first part of the cell which we'll discuss in a little bit detail, and we call it as plasma membrane. Okay, so both the prokaryotes and the eukaryotes have plasma membrane, which is a double layered like structure if you see, and it is made up of lipids that separates the cell interior from the outside environment, and this double layer is largely specialized lipids. We call it as phospholipids, which we call it as phospholipids, which is phosphate bond and the lipid molecule. Okay. Now, our actually body is composed of 70% water. So here within the blood vessels, the blood is there, and if you see the cell inside also we have water, and the within the two cells, the space is there. Within that also there is water. So there are lots of water. So how this lipid phospholipid layer will look like? Because they, if you see here the structure, phospholipids have a bilayer. So why there is a need of bilayer? Now I will explain you that part. Why not single layer? What will happen if it has a single layer? So we will keep in mind that our body is composed of water almost everywhere. So it is 70%, but if you see the structure, everywhere it is the water, and the plasma membrane is this border. Okay. Now, phospholipid is made up of the hydrophilic head, which we call it as water-loving, okay, phosphate head, along with the two hydrophobic tails, which is water-fearing. So this part is water-fearing; they cannot resist water at all, and this is a hydrophobic philic. That means they are water-loving, okay. And so this part is a fatty acid, which call it as tails. And phospholipids spontaneously arrange. So if you see the structure, they spontaneously arrange themselves in a double layered structure with hydrophobic tails pointing inward. So this is the hydrophobic, which cannot resist water, and their heads are facing outwards. And this energetically favorable two-layer structure, we call it as phospholipid bilayer, and hence it is formed like this, because outside there is water, inside there is water, and the hydrophilic are water loving and they are facing the water, and hydrophobic layers are not facing the water. Okay. Proteins are also important component of the plasma membrane. We'll see here that, and serving as a channel, so as receptors. So if you see here, they act as a channels. So in between there are glycoproteins. So these are the proteins, and here these helps in the transportation process. Okay. And different types of lipids like cholesterol is also there, which may also be found in the cell membrane and affect its fluidity. Now we'll discuss this fluid mosaic model of the plasma membrane, and I have shared one YouTube link. I thought it is very beneficial for those who want to go into fluid mosaic model in a bit detail. But basic thing of fluid mosaic model is that the structure of the plasma membrane is a mosaic of components. Mosaic, you have seen the mosaic floor, right? How it is located. So it is just like that, including phospholipids. So these are the phospholipids. They contain cholesterol, proteins, carbohydrates, etc. And according to this model, there is a lipid bilayer, okay, in which protein molecules are embedded. So their proteins are just like in the mosaic format. And the lipid bilayer gives the fluidity. If you see here, it is very fluid, it is very flexible, so it keeps on moving. So the purpose of the lipid bilayer is that they are elasticity of the membrane is beautifully maintained. So that is the whole point of the fluid mosaic model. And also, if you see here, the model or the proteins helps in the two processes, the transport of various nutrients. And the small molecules can enter through diffusion, but for larger molecules, like if you want amino acids to enter, then it requires some active transport. So this will learn in due course of time in the third subsection of these cells. But for now, you just remember that this bilayer, this plasma membrane, plays a very important role in the transportation of molecules or materials inside and outside the cell. The second part is the cytoplasm. Cytoplasm is the second part that we'll discuss. Now, cytoplasm, we know that it is a jelly-like material that fills up each cell and is present in again both prokaryotes and eukaryotes, and it is enclosed by a cell membrane. So there is a cell membrane outside, and inside there is a cytoplasm. Now, the part of the cell referred to as cytoplasm is slightly different in eukaryotes and prokaryotes. In eukaryotic cells, what we know that there is a membrane and there are membrane-bound organelles. There is a proper nucleus, right? So, cytoplasm is everything between the plasma membrane and the nuclear envelope. So, the nucleus also have an envelope, right, in eukaryotes. But in prokaryotes, there is no envelope, right? There are no membrane-bound organelles, and which lacks nucleus. Cytoplasm simply means everything, whichever is found inside the plasma membrane. So cytoplasm is that, but for eukaryotes, it's still the nuclear envelope. Okay, and because within the nucleus, we call it as nucleoplasm. So some material component, there is a difference in the jelly-like. So both are jelly-like materials, but within the nucleus, we call it as nucleoplasm, and outside the nuclear envelope, we call it as cytoplasm. Okay. Now, the one major component of the cytoplasm in both prokaryotes and eukaryotes is a gel-like cytosol. We call it as water-based solution that contains ions, then small molecules, macromolecules, etc. In eukaryotes, the cytoplasm also includes the membrane-bound organelles, right, which is suspended within the cytosol. Now, even though the cytosol is mostly water, it is 70% of the cell volume, but it is mostly water. It is a semi-solid gel-like consistency. So, I don't know, it is not paste, just toothpaste. You have seen that it is not as thick as that, but it is more consistency in the liquid. It is more than thicker liquid, you can say. It is more viscous because of many proteins suspended in it. And the cytosol contains a rich broth of macromolecules, smaller organic molecules like glucose and simple sugars also are there. Okay, and they have minerals like sodium, potassium, calcium, and other elements, and they are used in many processes like protein synthesis, and they all process takes place in the cytoplasm. So this is very important. Now, the function of the cytoplasm is just categorized as follows. Like they have the main three functions of energy, providing energy, site of energy, not providing energy, but site of energy synthesis, storage, and manufacturing. It contains other organelles which store and produce energy, because the energy is mainly produced by mitochondria, we know, but with the help of cytoplasm acts as a media to produce energy. The cytoplasm maintains the shape and consistency of this cell, right, and provides suspension to the organelles. And all the functions of the cell expansion, like growth and replication, are carried out in the cytoplasm of the cell. Because though the DNA replication occurs within the nucleus, but within the nucleus, they produce different processes like transcription and translation, and finally the translation, which is protein synthesis, they occur within the cytoplasm, because in that the ribosomes plays a very important role, and ribosomes are not within the nucleus, they are within the cytoplasm. Okay. Now, nucleus. Now, before starting that, suppose that you have a very precious piece of information. Let's imagine that this piece of information is a blueprint, okay? And in fact, it's not just any blueprint of a house like a car or even a jet. It's a blueprint of the entire organism, okay? For example, you. So you must think that how important is it, right? And so you probably want to keep this information very valuable and secured. So this information is very important, and in fact, that's exactly what eukaryotic cells do with their genetic materials, which is the DNA, and they place it in a membrane enclosed part. We call it as nucleus. So it is very, very secure. It has an envelope. It has different composition, so it is a secured information which stores the information about that particular cell. Okay. Now, eukaryotic cells never leaves the nucleus. This nucleus stays within the cell, of course, and it's transcribed into RNA molecules. So here the transcription process takes place. So this is the nucleus. It is just magnified here, and this is the transcription happens within the nucleus, and then we call it transcription is a process where actually the DNA, whatever is the genetic material within the nucleus, they form the RNA. We call it as messenger RNA, and this messenger RNA actually then transfers it moves out of the nuclear membrane and goes to the cytoplasm, and then the protein synthesis takes place within the cytoplasm. But this nucleus plays a very important role in the process of transcription, okay, because they form the RNAs. They form various RNAs like messenger RNA, transfer RNA within the nucleus, okay. So this is one fun fact that we can discuss, like even most cells have a single cell nucleus, right? We know that the nucleus of a single nucleus. A cell have a single nucleus, but for some cells like skeletal muscle cells, so they have which play a very important role in forming your biceps or triceps or quadricep. They are actually multinucleated, means one cell have more than one nucleus. So these type of cells doesn't have one nucleus, okay. Now the nucleus, the plural we call it as nuclei, they houses the cell's genetic material. We know that this DNA, and it is also the site of synthesis of ribosomes. So the ribosomes, which play an important role in protein synthesis, they are actually formed within the nucleus. So it is very, very important, right? And this inside the nucleus, the chromatin, that is the DNA, whatever is there, they are surrounded by proteins. And the gel-like material is there. We call it as nucleoplasm, not a cytoplasm. We just now mentioned, right? And enclosing this nucleoplasm is a nuclear envelope. So this blue is called the nuclear envelope, and an inner membrane. So they have also just like a phospholipid layer, they have also two. It is a bilayer, and each of these membranes contains two layers of phospholipids arranged with the tails pointing inward and the head pointing outward. So this is an outward, this is an outward. So there will be the head of the phospholipids, and inward will be the tails. There is a thin space between the two layers of the nuclear envelope, and this space is directly connected to the interior of the membrane. So if you see here, this space is connected with the endoplasmic reticulum. So this way it is actually connected to the endoplasmic reticulum, and here the mRNA goes to the endoplasmic reticulum where the ribosomes are situated and protein synthesis takes place. The nuclear pores are the small channels. These are the pores that actually span the nuclear envelope and let the substances enter and exit the nucleus. So the transportation also takes place. So this is the entire details of the nucleus, how it looks and how it functions. Okay. Now this is a picture of the DNA. So if you see the DNA and magnify it, it looks like this. The DNA strands of chromosomes are bound to structural proteins. So it is not only DNA. DNA, if you know that there are nucleic acids, there are 4 types of nucleic acid that form the DNA. That is adenine, guanine, cytosine and uracil, right? And they are not only bound to each other, they have protein surrounding the DNA. And this protein we call it as histone. So please remember this name. This is very, very important. So this histone, which is the protein associated with DNA, they form the chromosome. And within one nucleus, there is only one DNA. Though they are double helix, so there is one chromosome and one DNA. Okay, but for human cells, we know that one chromosome equal to one DNA, but there are 23 chromosomes, right, from the parent. So the human cells, for human cells, we have 42 chromosomes, and each chromosome have one DNA. So one chromosome, one DNA, and 23 chromosomes from mother, 23 chromosomes from father. So they form the human nucleus. So this is just an extra information that may be useful for later on. Yes. So what is the function? Just briefly I have mentioned, though we have discussed that they control the cellular functions, they control the synthesis of structural proteins, they contain the genetic information for reproduction because the replication happens within the nucleus, right? And even the formation of RNA happens within the nucleus. And also they play a very important role in our development because all the blueprint is within the nucleus and our behavior, okay? So all the genetic information is stored within the nucleus. Ribosomes are the fourth part of the cell that we'll discuss in a bit detail, and they are the molecular machines for photosynthesis, for protein synthesis. Okay, and a ribosome is made out of RNA and proteins, and each ribosome consists of two separate RNA protein complex. Okay, one is called the smaller subunit and one is called the larger subunit. This larger subunit sites on the top of the smaller. So smaller is in the bottom and the larger is on the top with an RNA template sandwiched between the two. So this in between there is an RNA template. Okay, this RNA name is messenger RNA, and this messenger RNA is formed in the nucleus. Okay, this gets moved through the nuclear pores and comes to the cytoplasm or on the endoplasmic reticulum where the ribosomes are situated. Okay, and this ribosome simply looks like a hamburger or a burger. Simply, you can think that that burger breads are there and some veggies or cheese are there here in between. Okay. And in eukaryotes, actually, the ribosomes get their orders for protein synthesis from the nucleus, where the portions of DNA or the genes are transcribed into making the messenger RNAs. Okay, we call it as transcription, which happens within the nucleus. And here it is called translation, which happens in the cytoplasm. So protein synthesis happens in the cytoplasm. Ribosome plays an important role in the translation, not transcription. Okay, and here the prokaryotes lack a proper nucleus. We know that, right? And so their mRNAs are transcribed in the cytoplasm and can be translated by the ribosomes immediately. So this is the case of the example of the eukaryotes, but for prokaryotes, since there is no envelope of nucleus, right? So everything happens in the cytoplasm. Okay, now this is just an example where the larger subunit is there and the smaller subunit of the ribosomes are there, the messenger RNA is there. Okay, and this is also another type of RNA, we call it as transfer RNA, and here the peptide bonds are formed. These are the two amino acids. Okay, so it's 1, 2, 3, and here the amino acids form the protein chains. So this is how the proteins are formed. We are made up of mostly proteins, right? Every cell have a specific function. So proteins are very, very important, which are synthesized. We have only 20 essential proteins we know, and rest all are non-essential. That means they are formed within the cell. So this is how the non-essential amino acids or the proteins are formed. These are the amino acids. This is of course one amino acid, but a chain of amino acids forms a protein, and in between we call it as peptide bond. So two amino acids, there is a peptide bond. Okay, now eukaryotic ribosomes may be either free, they are freely within the cytoplasm, or they are floating around in floating around in the cytoplasm, or bound, that is they are attached to the endoplasmic reticulum. Okay, now in this diagram we see that they are free ribosomes, and in this diagram that means they are attached to this one, endoplasmic reticulum. We call it as rough endoplasmic reticulum. Okay, the one fun fact is that the pancreas, for example, we know that it is a part in the digestive system, and it is responsible for producing and secreting a large amount of digestive enzymes, right? And so these pancreatic cells actually make these enzymes. So the cells of the pancreas make the enzymes. An enzyme is nothing but a protein. So we can say that the pancreas have unusually high number of ribosomes because they require more proteins, they require more to form more enzymes, which helps in the process of digestion. So see, we are moving from micromolecule to macromolecule. So the each cell is producing the enzymes or the proteins, and which helps in the process of digestion. Okay. Now the second last cell part that we'll be discussing is mitochondria. Mitochondria and chloroplast, we'll discuss it together. But firstly, we can know that your body is made of cells, trillions and trillions, right? And so you may know that the reason you need to eat food, for example, veggies, broccoli, cauliflower, spinach, so that you get energy from them, right? And play sports, study, work hard, breathe, respire, etc. But what exactly happens, right? Just think in a micromolecule perspective, and which turns the food energy of your, for example, spinach you eat or broccoli or cauliflower you eat into the form that your body can use. So the energy from a plant is translated to your energy to of the body that you need for doing various activities. So mitochondria are nothing but they are the powerhouses of the cell, breaking down fuel molecules of the food. So fuel is nothing but the food, and capturing energy in cellular respiration. Okay. Now the answers to this question have a lot to do with the two important, that from where and how it is formed, that we eat veggies and we get a lot of energy. So actually the two important organelles that play a very important role here is the chloroplast and the mitochondria. Why chloroplast? Because chloroplast are the sorry, the organelles found in the veggies that you eat, right? They have a cell. So the veggies like spinach or broccoli or cauliflower, they have a cell which contains chloroplast. And along with these other, just like other plants, and they capture the light energy and store it as a fuel in the plant's tissues. And mitochondria are found inside your cells. So we don't have a chloroplast, we are not plants, and so we have mitochondria, and here we have the convert. They have the power to convert the energy stored in the molecules from the spinach or other fuel molecules into the form of energy that we can use, like respiration, running, walking, etc. So there is a conversion of energy. Now chloroplasts are found, this is the detailed structure of the chloroplast. They are found only in plants and photosynthetic algae, of course. So the humans and other animals don't have the chloroplast, you already know it. The chloroplast's job is to carry out the process called photosynthesis, which happens in the form which takes place in the presence of light. So in the photosynthesis, what happens? The light energy is collected and used to build sugar from the carbon dioxide, right? And the sugar produced in photosynthesis may be used by the plant cell, okay, or may be consumed by animals. They eat like humans. And the energy contained in this sugar is harvested through a process of cellular respiration, which happens in the mitochondria again. For both the plants and animals. So both plants and animals have mitochondria. Mitochondria is common, but chloroplast is common only to plant cells and the algae, photosynthetic algae. So there is an energy transfer taking place. Okay. So chloroplasts are the disc-shaped organelles found in the cytosol of the cell. So this is the cytosol. They have outer and the inner membrane, okay, with an intermembrane space in between them. And within this membrane, discs known as the thylakoids are there. So if you see here that there is a double membrane shaped, and within the membrane, the discs known as thylakoids are present, and they are arranged in interconnected stacks called the grana. So we call them as grana. And the membrane of a thylakoid disc, if you see here, this is the stacks of grana, and the membrane of a thylakoid disc contains a light harvesting complex which contains the chlorophylls. The chlorophyll pigments are there, and the space inside the disc. So there is a space here where the grana is not there. We call it as stroma. So we name it as stroma, and this is the fluid. So this is a fluid, just like a gel-like structure, gel-like material. Okay, and so this is the detailed structure of chloroplast. So questions may be asked that where the chlorophyll pigment is there. So your answer will be grana and not stroma. Okay, because grana the thylakoids are in the form of a stacks, and the whole structure we call it as thylakoids. Okay. Now mitochondria in detail, we can see that they are often the powerhouse or the energy factories, right? This job is to make a steady supply of the energy molecule. What is the energy molecule? We call it as ATP, or adenosine triphosphate. So the more detailed molecular composition of this energy is ATP. And this is the this ATP is generated using chemical processes, and such as and their fuels is the sugars that we take from the vegetables or the food that we eat. And they also a process going on in the as a cellular respiration. Okay, and many of the processes of cellular respiration takes place within the mitochondria. So the mitochondria are suspended in the jelly-like cytosol. Of course, they are present within the cytosol, and they are oval-shaped mainly, and they have a two membrane. So just like chloroplast, they have also a two membrane. One is inner and one is the outer membrane. And the inner one which have inward protrusions. So if you see the detailed structure of this inner membrane, so they have an inward protrusions. We call it as cristae. So this projections within the inner membrane, we call it as cristae. Okay, why the cristae is important? Because they increase the surface area surrounding the whole organelle. Okay, and we will see that this surface area is important again for the energy production. So more surface area means more energy production. Okay. Now this is a more detailed structure. Like if you see the structure, they have more compartments. Just now we discussed the intermembrane space is there, the cytosol is outside, and here there is a fluid-like structure. We call it as matrix. And here the other also metabolic reactions takes place, like cellular respiration. And this is the inner membrane. Inner membrane is important for the respiratory chain proteins are present, folded into multiple cristae. So these projections are called cristae, while the cellular respiration takes place. And this is the outer membrane, which is permeable to some ions, not all. So they are selectively permeable and to some small ions and small molecules. So the main function is, of course, the aerobic metabolism. Aerobic means in the presence of oxygen. And secondly, it is energy production through oxidative phosphorylation. This is just a process name. Don't worry about it. And the third main function of the mitochondria is to maintain and replicate their own DNA. So yes, the mitochondria and chloroplast have their own DNA, just like nucleus. Fun fact that is good to understand and remember, that mitochondria, just like human cells, vary in the number of mitochondria varies from cell to cell. For example, the mitochondria is very high in number in muscle cells, because muscles we require energy. We have to move and contract and retract, relax. On the other hand, the red blood cells, or RBC, they don't have any mitochondria because they are highly specialized for oxygen transport. Okay, so you see the difference, like the mitochondria based on the function of the particular cell. The number of mitochondria or the number of ribosomes varies from cell to cell. Now, the origin of mitochondria and the chloroplast. This is also a very important thing that is important for your examinations point of view, entrance examinations point of view. So if you think the both mitochondria and chloroplast contain their own DNA, right? Just like the nucleus and as well as ribosomes. So why these organelles need DNA and ribosomes when there is DNA in the nucleus and ribosomes in the cytosol? So why they need their own something on their own? So actually, there is a strong evidence points towards this theory. We call it as endosymbiotic theory. And this symbiosis, what is symbiosis? Symbiosis is a relationship in which organisms from two separate species they depend on each other and they help each other. Okay, endo means within, and a specific type of symbiosis where one organism lives inside the other organism, which are close to each other. Okay, so this is an example. Like it is believed that the ancestral eubacteria or the true bacteria have engulfed the ancestral eukaryotic cell. I'm sorry, the ancestral eukaryotic cell, which is different from the prokaryotes. So previously, whenever the eukaryotic cells were formed, they had engulfed a prokaryote. So there is a proper membrane bound structure and they have engulfed a prokaryote or a bacteria, and hence the mitochondria have formed. So here it is modern heterotrophic eukaryote. Within that, the mitochondria has developed into a separate organelle due to the evolution. And the second theory is that the descendant consumed photosynthetic bacteria that evolved into chloroplasts. So the bacteria, we also have seen that there are various types of bacteria. So maybe the eukaryotic cell, ancestral eukaryotic cell, which have engulfed, which have eaten a photosynthetic bacteria, have transformed into a chloroplast. So this is a modern chloroplast, and this is a modern plant cell, and this is a modern animal cell. But previously, both were different. So it is believed. This is believed or proposed based on the endosymbiotic theory. And the reason of this belief is that mitochondria and chloroplast have their own set of ribosomes and own set of DNA. Okay. Now this is a set of just example. You can pause the video and take a note, just a small note of various organelles and their function in brief. And this is also a mind map that may be useful for you. You can draw in your notebook and can refer during or while the examinations. Okay. Now we will solve some problems within the class time, which are in the form of MCQ, which will help us to clarify our concept.